So we know that Alzheimer's disease is biologically heterogeneous, and a strong indication for that is the notion that there's lots of genetic variants implied in Alzheimer's, and then most of them only have very small effect sizes. So it's the specific combination of those genetic variants that kind of determine what type of Alzheimer's disease you will be developing. And so there's many possibilities of these different combinations. And we were thinking that perhaps the best way to study this disease heterogeneity in patients would be with proteomics analysis in the cerebrospinal fluids. So that's not really the subtypes yet, but a bit of the background. So this is something that we did previously. We had a big sample of Alzheimer's disease patients, which we defined by the presence of an amyloid marker that had to be abnormal across the clinical spectrum. And if you use a data-driven clustering algorithm, you can discover subtypes if they are present in the data. And we found that there were at least 3 different molecular subtypes that showed really distinct cerebrospinal fluid signatures. So one of those subtypes showed increased levels in the CSF of proteins associated with neuronal hyperplasticity. And this correlated also with very high tau markers. For example, an amyloid metabolism. So that all seems to be tied together. Another subtype showed the opposite relationship. So they showed abnormally low levels of those plasticity markers, and also abnormally low tau, or normal tau, and abnormally low amyloid processing. And that subtype showed increased levels of proteins associated with the blood-brain barrier. So these proteins were proteins that you usually see in the blood, and we found them in the CSF, which was indicating that the blood-brain barrier seems to be leaking in these individuals. And then there was a third subtype, and that subtype was a bit more similar to the first one, but they also showed indications of innate immune activation. Because there's these different, very distinct molecular processes being associated with the Alzheimer's subtypes, we think that this means that they probably need personalized treatment. But finding patients and selecting patients and matching them to the right trial and the right drug that requires screening procedures, and then a lumbar puncture might be a bit impractical. And this is a reason why we were studying this time whether we could also detect these CSF-based subtypes by looking into blood proteomics, because obviously blood is easier to draw. And we first take as starting point proteomics, because then you have really a large set of proteins, and you increase the probability that if we can see the cerebrospinal fluid signature in the blood, that we could make such a classifier or a subtype detector. And then maybe in the future, we can reduce the number of proteins and make it even easier to screen people. It is super interesting because the CSF molecular subtypes, the processes that are being distinctly dysregulated in the cerebrospinal fluid, we also see that in the blood. And if we then, so that was not obvious at the start. It was not obvious at the start, but the proteins deregulated really were enriched for the same processes. So that was the first step, to see whether or not we could really identify those CSF-based subtypes in blood. But then you still have a lot of proteins. So the next step that we took was to see whether you can also use like the top 5 proteins and then get a good accuracy of classifying the correct subtype or predicting the correct subtype. And we found that if you train such a classifier, which is a random forest, and I can explain a bit about that later, that we were able to detect each subtype with an average accuracy of about 75%, which is pretty good because we looked at we just let the data speak, so we didn't a priori select the most distinguishing proteins on the panel. And so this is pretty good news that if we look into the data a bit more, that it will be possible to perhaps increase these accuracies as well. Well, we only looked into one cohort, and to make the classifier, we did train it on a subset of the sample and left some people out so we could really test whether they could predict new cases. But still, it's just one cohort, and then the sample sizes are relatively small. So I'm now starting up new collaborations to look into larger cohorts to see, especially cohorts that have combined blood and CSF proteomics, to further validate such a classifier. And then in the future, we could look also into even larger data sets with more and more blood proteomics because it is easier to obtain. And then study, for example, how often a particular subtype occurs in the population. And then we also get a better idea of the subtype specific prevalences, and that will help with developing strategies to select and recruit people for trials if we find subtype specific drugs.